What is the contractile unit of a muscle called? Skeletal muscle. Sarcomere. What is sarcolemma? Covering of the muscle fiber. 5 yes. Roll 6. 7. What is the most important ion helping in muscle contraction? Calcium ions. Where they are coming from inside the muscles, where they are stored, where they are coming when muscle contraction is occurring, where are they coming? So I think you should have, and I studied about muscle contraction. Roll 7. What is end plate? Roll 8 you. What is end plate potential? All these will come as question. Each one of these objective questions, MCQ questions, short questions, whatever. Normally not long questions. Normally, except earlier there were 7 marks, 10 marks questions about steps of muscle contraction. Now excitation, contraction, coupling, short note, everything, whatever has been asked, I just asked now. Every word can come as a question in the theory exam. Excitation, contraction, coupling is electrical response to be converted to mechanical response. Now stimulation what is the chemical role? 9. What is the chemical present in the neuromuscular junction? So what is happening? The nerve is stimulated, muscle contraction is occurring. We study it experimentally in the usually an amphibian, a frog. Amphibian study ta kora hoy. Which muscle and nerve are chosen? Sciatic nerve and gastrocnemius muscle. So everybody open whatever books you have, practical books. AK Jainer boita kar ache? Kar kache ache? Anybody? Ja boi ache setai kholo. Practical book. Not this is practical or theory? Theory, practical er boita kothay? আছে কারোর কাছে? কারোর কাছে নেই? And কিছু বই তো আছে, প্রত্যেকদিন তো বই রাখতে বলা হয়েছে। Does anyone have this? AKJ and practical book? Nobody? কে কোনটা ব্যবহার আছে এখানে? Which edition? Okay, open page 77. '79 actually. '79. Page 79 তোমার আছে? ওকে, হ্যাঁ ওইটা খোলো। নার্ভ মাসল প্রিপারেশন, অ্যাম্ফিবিয়ান ফিজিওলজিটা খোলো সবাই। ওখান থেকে দেখো রিড আউট। ফ্রগস আর leave the introduction to study the nerve muscle. Read out that part. To study the nerve muscle preparation, frogs are preferred because AKJ খুলছো তো? Page 79. That is not page 79. Yes, if that is page 79, see a living tissue. See the headline? দেখেছো হেডলাইনটা? আরে পরে ছবি তুলবে, ওটাকে দাও। তোমার কাছে তুমি কোন বই ইউজ করো সেই বই থেকেই পড়বে। একটাই বই থেকে পড়তে হবে তার কোনো মানে নেই। ওখান থেকে পড়ো ওটা। ফ্রগস আর প্রেফার্ড ওই জায়গাটা থেকে পড়ো। জোরে জোরে জোরে। Frogs are the preferred animals in nerve muscle preparation because of the following reasons. So it is cold-blooded animal, that is one advantage. Okay, and they are absolutely harmless animals and available almost all over there. In our countries, maybe in dry weather a bit less, but in moist weather, dry weather is very less in our countries. And now, if you go and try to find it in Rajasthan, you will not find it in the deserts. But more or less, most in our country specifically, it is available. Okay, and then yes, that is the first one is. So first one is this is a cold-blooded animal. Then second one is availability. Third one is viability. Outside viability, and viability is better, and last one is they are easy to handle. So as they are covered in the nerve-muscle preparation is easy to prepare. So process of experimentation is also easy, availability is also easy, viability also easy. So it is chosen. In your syllabus, amphibian physiology is brief. Still, some parts are somewhat difficult. Cardiac part is a bit difficult. In the earlier years, we are not keeping it. Once upon a time, it was not taught in the MBBS syllabus. Now, NMC has specifically mentioned amphibian nerve muscle and cardiac experiments are to be discussed. However, we are not going into actually dissecting a frog and all. That is not in the syllabus. Even in the MD syllabus, it has been practically scrapped because due to ethical issues and all. So it has been practically scrapped. So earlier there were the things we had to do the total experimental setup during the, that means the MD examinations, and show it to the examiner and all. That has been, I think, scrapped in most institutions. So there will be probably 5, maybe 10, but I think not more than 5 marks from your exam point of view in your exam. Okay. So out of 100 marks in the practical exam, 5 marks will be from amphibian physiology. We will discuss over the maybe the next 2 weeks, maybe 2-3 weeks. 2-3 weeks means each group, that means we will have 3-4, maybe 5 classes on amphibian physiology maximum. So 5. So that is in brief. There are some charts which have some, these are not graphs, just some diagrams which will be supplied during the exam, and you have to explain. Questions will be asked. It is nerve muscle part is related to nerve muscle physiology. The cardiac part is related to cardiac physiology. So, and there will be some theoretical questions, and will be asked to explain the diagram. There will be some theoretical questions. So whenever this nerve muscle preparation is studied, there is an experimental setup. We are not going into the details of it. Just know there is an induction coil. It is called the Dubois-Reymond induction coil for this electrical impulse to be given. Electrical impulse you have to give, Dubois-Reymond induction coil, electrical impulse to the nerve, so that the muscle contraction is seen. A lever arrangement is made, and there is a stylus for writing, which is on a rotating drum. It is making during the muscle contraction, it is making a that is impression. That impression is what we study. We are representing as these diagrams, which you will find in the, we have charts. So which you will find in the chart. So that is called that arrangement where this impression is made. That is called the rotating drum. That is called a This is a kymographic representation. That is called a kymograph. So the Dubois-Reymond induction coil, the electrical impulse is given to the nerve, and in the reading is on a kymograph. It is a lever arrangement. There is a mounting apparatus has to be there where the nerve and the muscle So it is, which bone is there? Sciatic nerve, the femur sciatic nerve supplying the gastrocnemius muscle. Where is the situation of the gastrocnemius muscle? It is the one of the cuff muscles. So and we study the contraction. You can see the muscle contraction, brief contraction, and the stylus moves, and a contraction is recorded. That means, so it is seen that whenever there is the muscle, as the stylus is moving, when the contraction will be at its peak, this impression which will create, it will be a something like this. There is a baseline, starts like this, and impression is created like this. It looks like as if similar to a similar in diagram to an action potential. Similar. That is, this is completely different from action potential. Similar in diagram. এই রকম একটা দেখতে হয়. So this is when a single muscle contraction is recorded, this is the height of contraction. So it is seen that after the muscle contraction, when this diagram is produced on that kymograph, just after, when the stimulus is given, electrical stimulus, just after some time, but that cannot be recorded, that is very short, 1/100th of a second or less. So that is very short, that cannot be humanly, that cannot be recorded. So there is a time tracing curve. Humanly it can be recorded, it is 1/100th of a second or less, but actually the muscle contraction starts that very 0.01 second maybe after the stimulus is given. So something is recorded like this. This is called a simple muscle twitch, as it is called a twitch, as it is a one single exposure, after the stimulation, one single contraction was that is recorded. So it is called a simple muscle twitch, or short sometimes it is called the SMT, or the single muscle curve it is also called, as it looks like a curve, it's called the single muscle curve. Now this period actually, which cannot be recorded humanly, but it starts just after it is recorded in this tracing, this is called the latent period. This is the height of contraction, this is called the contraction period, and this is called the relaxation period. Tuning forks are used for this time tracing, so total time. So that is, again, this is, all of this occurs in small fractions of seconds. So this is time tracing is required, humanly it is not possible, and there is one curve after this, a small curve after the actual curve. Actual, I am telling, the contraction relaxation, that is not due to muscle. What happened? We were talking in the class. So this actually occurs, it is seen after the actual curve. Actual we are talking, that is the contraction relaxation. The small one which is recorded is called the physiological curve. Now this name is somewhat a type of a misnomer. It has nothing to do with physiology, that is the contraction and relaxation phase. It has nothing to, contraction relaxation has ended. That is just due to the inertia of the lever. Muscle contraction has stopped, the lever is still rotating. Rotating means, sorry, the working, due to the inertia of the lever, the experimental lever setup, that has not stopped, that the stylus is on there, that just draws that small curve. It was moving like this, the lever had moved like this, so it draws that small curve, that is called the physiological curve. Although physiological curve has nothing to do with the actual this muscle physiology, contraction and relaxation. So somewhat a type of a misnomer, but if you are asked what is the physiological curve, what it is called, why is it caused, physiological curve is this. You have to show the graph, show the diagram. This one is part is called the physiological curve. This is just due to the inertia of the lever. This has nothing to do with the physiological contraction of the muscle. So whatever books you have, just turn the page and see the simple muscle curve. This one, A to B is latent period, B to C is contraction period, C to D is relaxation period, and after D you can see the small physiological curve. Okay. So this is in case of a simple muscle twitch. Sorry. This is a simple muscle twitch. Now what happens? At higher temperatures, if the muscle is exposed to higher temperatures, this latent period gets shortened. So what is the effect of temperature on these muscles? So enzymatic reactions are faster. Okay. So this latent period gets shortened and the contraction amplitude is more. Amplitude of contraction, that height of contraction is more. Okay. So this is in case of a muscle at a higher temperature. Say this is normal versus at 40 degrees centigrade. Okay. At lower temperatures, what happens? Again, the latent period is increased and the height of contraction is less. So this may be combined, this thing. It is written 10 degree centigrade, maybe 12 to 14, 16 degree centigrade, okay, or lesser. But if temperatures are further raised, if it goes beyond a certain viable, say 50 degree centigrade, 60 degree centigrade, or it is brought down to 0 degree centigrade, then nothing will be obtained. Actually, the muscle can't contract. Physiologically, or contraction is possible. So these curves won't be obtained. It is not like that. So it has to be within a certain range. 10 থেকে 40 মধ্যে changes, এরকম temperature 25, 26, 29, 30, maybe up to 35, 36, the normal simple muscle curve. Temperature change holo, it's a simple muscle curve. It is still a simple muscle curve. But if we compare with one at 25 degree centigrade, then the amplitude of contraction is more, latent period is less. This sometimes in the exams it is combined in a single diagram. If it is there, I will show you, otherwise you will get it in the books. Ekta single diagram e dewa thake তিন রকম. So one is normal, you have to identify which is normal, which is at higher temperature, which is at lower temperature. Very simple, you will see the amplitude of contraction and the latent period. যেটা মাঝামাঝি থাকবে, that is at room 25 degree centigrade, or you are just asked which of these is at the room temperature, which of these is at a very high temperature, which of these is at a considerably lower temperature. So you just identify and tell. The latent periods will be smaller in case of the higher temperature, larger in case of the, and larger than what, the, which is for the room temperature. So you can just identify. Here it is not there, all are separate, so it can be combined in a single diagram. And this below line, these curves are for time tracing. Just use, utter the word time tracing, this below one, for time tracing. Whenever this is say combined in a very short period of time, strength of the stimulus can be experimentally varied, the strength of the stimulus. So whenever it is combined very slowly, whenever it is combined, say a second stimulus is given in the just after the first. Just after means, now just after can vary, okay. So just after, it cannot be given humanly. Before that, we just show this one. So it depends on the strength of the stimulus. If an increased stimulus is given, there is increased height of contraction. Now this is just represented here. So the experimental setup is slightly varied, so that the normal muscle curve, this simple muscle twitch, without not recording it. Here experimental setup, there are different these structures in the instrument. There is the primary key, there is the secondary key. You don't need to go into the details of the setup. Does no Dubois-Reymond induction coil is used. So we can vary the current, okay. So there is something called the make current, break current. We don't need details. So when we can vary the current, the strength of the stimulus can be varied. If the strength of the stimulus is more, the height of contraction will increase. Now this height of contraction again can increase up to a certain limit. Ei je height of contraction ta jodi amra korte jai, so if we give an increasing strength of stimulus, it can become like this. So height of contraction can be increased, but it will be seen that after a certain stimulus, if stimulus is still increased, height of contraction does not increase anymore. This is just represented in this diagram, height of contraction. So that is the maximal stimulus. Above that, supramaximal stimulus, any stimulus. So what is the maximal stimulus? The lowest stimulus at which the height of contraction is maximum. After that, whatever stimulus we give, height of contraction is seen that is not increased. The physiological cause behind this is that muscle contraction occurs due to involvement of motor units, okay. So motor unit is that an area supplied by that this neuromuscular junction is not supplying, in the junction of the nerve and the muscle, there are these motor units. So all motor units, when they are recruited, so when the stimulus is increasing, height of contraction is recorded is more. How? Because these motor units are recruited more in number, but a certain time comes after a certain amount of stimulus, all the motor units have been recruited. Okay. Now it is no more, there is no motor unit left to be recruited. Then the height of contraction is seen does not increase anymore. So this is the effect of this is how effect of increasing stimulus is seen in experimental setup, and it is concluded the muscle follows the all or none law. Whatever motor unit is recruited, you have heard of the all or none law in the theory class? Whenever motor unit is recruited, either it will contract fully or it will not contract, and once all motor units are recruited, so it has contracted to its maximum. So it's not that say motor unit number 1 has worked, but it has not worked maximally. So either it will work full or it won't work. So some is left. So if that was possible, then what would happen? Then even after the maximal stimulus, say the previous motor units have now relaxed, and now they are again, that does not happen. So it is given strength of stimulus, one motor unit will be recruited only after another has reached its full limit. So once all motor units have been recruited, all are working up to the full limit, no more muscle contraction is possible. So just think it simply, again detailed experimental setup not required, and more than this won't be asked for your exam. This is more or less, this part of your syllabus is more or less for your exam purpose, that much deep. So deep understanding is not required, but basic understanding is required. This is due to the increasing strength of stimulus, increasing strength of the stimulus. Now if this increasing strength of stimulus is given repeatedly, say just repeatedly, before another one relaxation happens, another strength of stimulus is, another stimulus is given, then what happens? So this is the simple muscle twitch, whatever books you have, you will get it. This is the simple muscle twitch, okay. In the book by Dr. Akajan, it is on page 94. Then effect of temperature, here it is combined, effect of temperature, okay. Effect of temperature on page 97. You can mark the page if you like, there is no hard and fast rule, you have to use the same book, you will get it. But if this book you use, it is on page 97 and it is combined. So 26 degrees centigrade, 38 degrees centigrade and 16 degrees centigrade. Effect of temperature. Just one, I showed you this one. These charts will be given to you during the exam. And this was one which I was trying to tell. Chart is right now not available. So this is on that book, this is page 105. Just if you have other books, see the chapter Genesis of Tetanus. So what happens when it is increasing frequency of the stimulus, then what happens? Then it is the genesis of tetanus. After a certain time, all these overlap. So that leads to this is called genesis of tetanus. When complete tetanus, then the muscle has gone into a state of sustained contraction. So this is the diagram for a complete tetanus. Just now the chart is not over here, but it can be produced during the exam. So tetanus, so the stages are prepi, clonus, and then incomplete tetanus and complete tetanus. So all these, know a few words, only these few words you are expected to utter, not more than that. Mostly you will be given the simple muscle curve and asked to explain the latent period, contractile period and relaxation period. One thing is that if same strength stimulus is given, then what happens? Same strength stimulus, I think the curve is here. So two successive stimuli of same strength on muscle contraction. So it can fall in different periods of the first contraction. So if it is given within the latent period, this is a simple muscle twitch. If it is given within the latent period, then there may be a slight change, slight overlap. See the 6, 1, 2, 3, 4, 5, 6, okay. So see the first one, this is a simple muscle twitch, everybody can see, simple muscle twitch, and see the last one, it is similar. So that is a second strength. What we just explained or what we just mentioned is the increasing strength of stimulus and the amplitude is increasing. But if the same strength is given, same strength of stimulus at a later time. So if it falls within the latent period, especially the first half of the latent period, there is no change. It looks like it is overlapped, or it coincides with the first twitch. But if it is in the second half of the latent period, the amplitude slightly increases. Okay. If it is in the relaxation period, then the second diagram, then it looks like a muscle twitch, still a separate muscle twitch, but still sometimes it is seen the amplitude of the second is slightly greater than the first. Again, the cause is similar physiological cause, that some motor units have already been recruited, others are being recruited. But if it is falls during the contraction period and the relaxation period, there will be some change in the curve, that during the contraction period of the first one, a second stimulus is given. So the first one was due to cause a height of contraction, but now the height of contraction, actual height of contraction increases, and the curve looks something like this. This left one, the bottom one, left hand side. Okay, curve looks something like this. So if you are given this curve effect of two successive stimuli, maybe this is not written, so you have to say this is the effect of two successive stimuli, but the main principle, which is the most important one, is not written. It is of same strength. If it is of different strength, then the two separate, that amplitude is increasing, that is into more motor units are separately engaged, but still up to a limit, up to that limit when the motor units are, these motor units are still the availability of motor units, that is still the availability of the motor units. So just pass this one. So this one again, simple muscle curve. Also see the books. It will be better for you to see the books, because you will study from the books. Turn it after you have seen it. This is the effect of temperature, that is 40 degrees centigrade. देखो, this is the effect of temperature. এগুলো দেখো, সবকটা পাস করে করে দেখো। ছবি তোলার কিছু নেই, যে বইতেই পড়বে সেই বইতেই পাবে, দেখে নাও এটা। বাট এফেক্ট অফ টেম্পারেচার ইউজুয়ালি কম্বাইন্ড, এই যে এক্ষুনি যে বইতে যে ছবিটা দেখালাম, ওটা কম্বাইন্ড ভাবে থাকবে, লেখা থাকবে না, বলতে হবে যে দিস ইজ দ্য এফেক্ট অফ টেম্পারেচার, কোনটা ওয়ার্ম কোনটা কোল্ড, সবই এক জায়গায় নিও না, এটা নাও একসাথে, একটা দেখো তারপরে দিস ইজ দ্য, এটা কি, এই তোমরা দেখো ওইখানটা দিয়েছি, তোমরা দেখো এটা কি, এটা কোনটা, এটা হচ্ছে ইনক্রিজিং স্ট্রেংথ অফ স্টিমুলাস, যে হাইটটা বাড়ছে, ইনক্রিজিং স্ট্রেংথ অফ স্টিমুলাস। This is the effect of two successive stimuli of same strength. This is the strength of, একটা ওদিকে দিয়েছিলাম, increasing strength of stimulus, increasing stimulus, and another is same strength second stimulus. Both are different. এটা আরেকটা আছে, this is the simple muscle curve. অরেঞ্জ কালারটা তো বোঝানোর জন্য দেওয়া হয়েছে, কি বলছো না, অ্যাকচুয়াল দ্য মাসেল কার্ভ উইচ বি ট্রেসড যেটা হবে ওটা ব্লু কালার, অরেঞ্জ কালার গুলো এখানে বোঝানোর জন্য দেওয়া হয়েছে, এ বি সি পয়েন্ট গুলো বোঝানোর জন্য, যেটা ট্রেসিংটা হচ্ছে দ্যাট ইজ ব্লু কালার, অ্যাকচুয়াল ট্রেসিং যেটা ওখানে হবে, হ্যাঁ মানে সেটা এখানে ব্লু কালার, মানে ওখানে তো স্টাইলাসে থাকবে, ওটা একটা সাদা চক, বা চক নয়, ওটা চক নয়, সাদা এতে কালো ড্রাম কাইমোগ্রাফ, ব্ল্যাক এর উপরে সাদা এ দিয়ে করা হয় অ্যাকচুয়াল এক্সপেরিমেন্টাল সেটআপে। হোয়াট ফ্রিকোয়েন্সি ইজ ইউজড হিয়ার, টিউনিং ফর ফ্রিকোয়েন্সি অফ 100 হার্টজ ইজ ইউজড, 100 হার্টজ, এই এক্সপেরিমেন্ট গুলোতে 100 হার্টজ ফ্রিকোয়েন্সি ইউজ করা হয়, 100 হার্টজ, ওই টাইম ট্রেসিং এর জন্য। ম্যাক্সিমাম 10 না 5 নম্বর হয়তো থাকবে পরীক্ষায়, কিন্তু কোনো একটা কার্ভ দিয়ে একটু এক্সপ্লেন এন্ড থিওরিটিক্যাল কোশ্চেন্স। অনেক সময় এক্সামিনার যেটা করতে পারেন, যে ফার্স্ট পেপারের ভাইবা, শুনে নাও, পরীক্ষার জন্য বলে দিচ্ছি, ফার্স্ট পেপারের ভাইবা যে হয়তো নিচ্ছেন, সাধারণত পরীক্ষায় যদি এক্সামিনাররা এগ্রি করেন, অনেক সময় দুটো পেপার একসাথে নেওয়া হয়, বাট দ্যাট ইজ দ্য বেটার রেকমেন্ডেশন হচ্ছে ফার্স্ট পেপারের ভাইবা, সেকেন্ড পেপারের ভাইবা আলাদা আলাদা এক্সামিনার ফেস করতে হবে, and NMC is wanting that each student will face all examiners. I am saying on record, practically সব সময় সেটা হয় না, কিন্তু সেটাও to maintain uniformity. Each student, একজন 5 জন examiner আসেন, 5 examiners are coming, that means 3 are coming from outside, 2 are from inside. The rule is that at least at least 3 have to be from outside your institution, and 2 of them have to be from outside the university which you are affiliated to. They are working somewhere else under the college of some other university. These are exam rules. Now more number of external examiners can be there, but less numbers cannot be there. That is not allowed. So at least 3 from outside, 2 from the institution you are studying in, or maybe 1 from the institution you are studying, 4 from outside, and each student is, NMC is recommending that each student has to face all the examiners. Now Viva 1, Viva 2, if they are separate, then sometimes these 5 marks are combined with Viva 1, that is nerve muscle physiology যেখানে জিজ্ঞেস করবে কোশ্চেন, তার সঙ্গে সঙ্গে এই 5 নম্বরের পরীক্ষা হয়ে যাবে। দিস ইজ আ সেপারেট টেবিল। তোমাদের মানে অ্যাকচুয়াল এক্সপেরিমেন্টাল সেটআপের সামনে কোনো অ্যাম্ফিবিয়ান পরীক্ষা দিতে হবে না। সো সেখানে ইউ উইল বি গিভেন দ্য কার্ভস অ্যান্ড ইউ উইল বি আস্কড টু এক্সপ্লেন। কার্ভস যে এটাই থাকবে, তা কার্ভ যদি আবার করে দেওয়া হয় যেখানে এক্সামিনার চাইবেন, বা তারা যদি নিয়ে আসেন যে কার্ভস থাকবে যেখানে these things will not be written what it is about, just the curve will be there, nothing will be written. So if this is like this, what is point A? So point A is point of stimulus, point B is start of the contraction, so AB is latent period. Okay, so C যদি এখানে না লিখে এখানে একটা লেটার কিছু লিখে রাখে, সে কিছু অন্য কিছু লিখলো, S বলে লিখে রাখলো। So what is S? S is the amplitude of contraction, maximum height of contraction when the contraction is at its peak. Just go and study in your books today, তারপর আবার এটা রিভিশন করবে মাঝে মাঝে, তাহলে হয়ে যাবে। This is the effect of higher temperature, but in exam it will be combined, like the one we showed in the book. Usually it is combined, তারপর explain করতে বলা হয়। So this is the same strength, effect of same strength at different periods, same strength of stimulus, second stimulus at different periods, 2 stimuli at different periods, within the first half of latent period, within the second half of latent period, then within the contraction period, within the relaxation period. Jodi relaxation period hoy tale that will be a separate curve. This is showing the increasing strength of stimulus and height of contraction increasing. At maximal stimulus it will be the maximum contraction. Now when we give a higher stimulus, contraction is not, height of contraction not increasing, then we can define this stimulus, that is the lowest value of the higher stimulus, lowest value যেখানে তারপরে আর স্টিমুলাস বাড়িয়ে আর it is not increasing, that is defined as the, or that is designated as the maximal stimulus. Higher than those are supramaximal stimulus, যেখানে আর contraction করছে না. Physiological cause is recruitment of all motor units have been done, and this another is genesis of tetanus, trepiclonus tetanus. Tetanus এর chart এখানে নেই, you see the book. Trepiclonus, incomplete tetanus and complete tetanus with increasing frequency of this increasing strength of stimulus. Same stimulus নয়, strength of stimulus ও increase করছে and very high frequencies. So in nerve muscle some more charts are there. Effect of load টা বাকি থাকলো, effect of load and this genesis of fatigue. So again these will be revised, and এটা হয়ে আমরা nerve muscle Friday তে nerve muscle part same, maybe next week cardiac টা করে। So HOD স্যারও বলছেন যে এগুলোতে নম্বর খুব বেশি থাকে না, so you need not read days for it. More important is clinical, এখনো clinical এর CNS পুরো বাকি আছে। So this is, এটা একটু শর্ট, 5 নম্বর থাকবে, কিন্তু যেটুকু থাকবে সেটুকু ভালো করে প্রিপেয়ার করে আসতে হবে। অ্যান্ড অবভিয়াসলি নার্ভ মাসেলের এই যে কোশ্চেন গুলো, যে থিওরিটিক্যাল কোশ্চেন গুলো, এগুলো সঙ্গে সঙ্গে জিজ্ঞেস করা হবে। ওকে, সো রেস্ট 1 or 2 কার্ভস ইন অ্যাম্ফিবিয়ান নার্ভ মাসেল থাকলো, 1 না 2 3টে, সেইগুলো ফ্রাইডেতে হবে। অ্যান্ড তারপর অ্যাম্ফিবিয়ানের কার্ডিয়াকের কিছু কিছু আছে সেগুলো হবে। দেন উইল পাস অন টু সিএনএস। সিএনএস করাতে টাইম লাগবে। সিএনএস এর দ্যাট ইজ প্র্যাকটিক্যাল পার্ট। অন থার্সডে একটা একাডেমিক ছুটি আছে না? হ্যাঁ, ইন দ্য একাডেমিক ক্যালেন্ডার। তোমাদের তো ফিজিওলজি নেই প্র্যাকটিক্যাল। Thursday of this week and Friday of next week. Friday of next week. So still we hope that in these 2 weeks maximum তার পরের উইকের ফার্স্ট হাফটা লাগবে, we can do amphibian physiology. Amphibian শেষ করে ফেলবো। তোমরা পাবে, you will get this Friday, next Tuesday also at least 3 classes পাবে amphibian physiology, তারপরের উইকে গেলে 4। তাহলে নেক্সট উইক তোমাদের ফ্রাইডেতে ফিজিওলজি ক্লাসটা ওদিন হবে না, তাহলেও 3টে ক্লাস পাওয়া যাবে। থার্সডের ব্যাচেও 3টে ক্লাস পাওয়া যাবে, ওয়েডনেসডের ব্যাচে 4টে ক্লাস। সো অ্যাম্ফিবিয়ান, বাট দিস ক্লাসেস ওন্ট বি রিপিটেড। আমরা পরে টাইম পেলে তবে আবার করব। সিম্পল, রিলেটিভলি সিম্পল থিংস, বাট কার্ডগুলো মাঝে মাঝে দেখবে আর নার্ভ মাসেল ফিজিওলজি পড়াশোনা করবে, না হলে পরীক্ষায় উত্তর দিতে পারবে না। ওই 5 নম্বরে 3-4 নম্বর পেয়ে গেলে দ্যাট ইজ ওই 100-র মধ্যে আলটিমেটলি তত নম্বর বাড়তে থাকবে। এগুলো স্কোরিং এরিয়া। ক্লিনিক্যাল আর ভাইভা ইজ নট স্কোরিং এরিয়া। ভাইবার মধ্যেই এটা হয়তো জিজ্ঞেস করছে, তো এতে অন্তত কিছু নাম্বার। আর নার্ভ মাসেলের থেকে এই যে অ্যাসিটিল কোলিন আর যে রিলেটেড যে থিওরি, সেটা থাকলে এখানে কিছু নাম্বার ভালো পাওয়া যাবে। এখানে 5 নম্বর আছে, এটায় 3-4 পেয়ে যেতে হবে।